All right, getting into the next thing, we're going to talk about the skeletal muscle on a microscopic side. So we're going to briefly go over the anatomy again. I'm going to go through pretty quick because we're going to mainly focus on the actions that these components are taking. So I'll briefly remind you of them, and then we'll get into the actual nitty-gritty of how contraction is occurring. All right, so if we remind ourselves of all the components, we have the contractile proteins. These are the proteins that actually do the pulling in. They do the contracting part, and that's going to have two. There's actin, which are the thin ones. We see them here in green. And then myosin, which is in this weird beigey pink color, is the thick filament. We have regulatory proteins that are going to basically help control when actin and myosin can work together to contract. We have troponin and tropomyosin. Troponin is going to bind with the calcium ions, whereas tropomyosin is blocking the myosin binding sites. And when calcium binds, troponin and tropomyosin will change shape or change configuration so that those myosin binding sites become revealed. Some other things, those are really the four big ones I care about the most. But if we look at some other things and what they're doing, these are just for structure. We have titin. This is going to help the sarcomere go back to its resting length, basically to stretch back out after we've gone through contraction. Nebulin, which will maintain the alignment of the thin filaments. Alpha-actinin, which binds the actin and the titin together. Myomesin, which is going to be the M line in the middle, right? And then dystrophin, which is going to transmit tension to tendons. And so I'm not as worried about the structural ones. Quite honestly, I'm really worried about these big four, the actin, the myosin, the troponin, and the tropomyosin. Those are ones like you must know what those guys are, because we're going to talk about the cycle of contraction and how that occurs, and those four are major players in that. So let's talk about actin. Actin is also called the thin filament. It is going to have binding sites on it where myosin can actually grab on. So myosin can actually grab onto actin at these specific sites. And then the myosin will pull and kind of pull itself along actin, shortening the entire sarcomere. Actin does not work alone. It is associated with the troponin, which we see in this gray color, and then the tropomyosin in brown, so actin is in yellow. And so the actin has the binding sites, the troponin, the tropomyosin are going to be covering those binding sites unless calcium is present. The myosin, this is a much better color, this in this like peachy color is the thick filament, because in comparison it's a lot bigger. This is a dynamic protein, meaning it is a moving protein. It's going to cause us to move the sarcomere. And so we can call it a motor protein for that purpose. We will go through ATP cycles, right, to generate that motion. Each of those myosin heads are like these little golf club shaped pieces coming off. Those are the ones that are going to bind to actin to cause movement. And then the tropomyosin and troponin, they're regulatory, so they're not actually involved in the they're not directly involved in the movement between actin and myosin, but they will regulate when actin and myosin can bind. So when it's relaxed, myosin cannot bind to actin because tropomyosin is in the way. But when calcium binds to troponin, this forces tropomyosin to move aside. And when tropomyosin moves, now those myosin binding sites on the actin are open and myosin can grab on. So how do we organize the cells and whatnot? How does this muscle cell even look like? So you may see the terms muscle fiber and muscle cell used interchangeably. That's fine. It's the same thing. So this muscle fiber, right, that we see here, this is the muscle fiber. This is one single cell, or at least a snippet of that cell. Within that muscle fiber, within that cell, we see myofibrils, which are each of these kind of cylinders right inside. So each of these cylinders is a myofibril. We also have the sarcolemma, which is just another name for the plasma membrane. The sarcoplasmic reticulum, which is a modified smooth ER, right? It's going to be this blue component kind of weaving around each of those myofibrils. We have the transverse tubules, also called the T-tubules. If you say T-tubule, I'll know exactly what you mean. Those are running transverse. See how they're going kind of across, right, and around, right? So we have those, which is what's going to help basically communicate between the muscle cell and the neuron. It's going to be what kind of links us to the inside from with the action potential coming from the neuromuscular junction. The sarcoplasm is just the cytoplasm, just renamed. We have more myoglobin and glycogen in there in comparison to a cell from somewhere else. Myoglobin is like an oxygen storage molecule, kind of like hemoglobin. And then we have the triad, which is each side of that kind of sarcoplasmic reticulum, right, what we call the terminal cisternae of that, and the T-tubule in the middle, right? So that's why it's a triad, because it's 3 kind of sides. And then, like I mentioned, the sarcoplasmic reticulum, right, is rolling around each one of those myofibrils and is going to store calcium. And when it's time to contract, it will release that calcium to bind to that troponin. So the sarcoplasmic reticulum is just the smooth ER that's been modified. It's a major storage facility for calcium ions. It is continuous with that terminal cisternae, which then will join up and sandwich the T-tubules. And when a muscle fiber gets stimulated, right, we have an action potential that occurs. It triggers those voltage-gated channels, specifically in this case for calcium, so that calcium will diffuse from the T-tubule, right? It will then talk to the sarcoplasmic reticulum, causing more calcium to diffuse out, right? And that will be used in muscle contraction. I'm not super worried about some of these like extreme details, but you should know about the calcium stuff. Now, as a reminder of how muscle cells are put together, we have the 3 layers. We have the epimysium. This is like the outer lining of an entire muscle with multiple cells. The perimysium, which is going to be the kind of bundling a few of these muscle cells together. So like here we see like they're kind of packed together. And then individually, we have an inner lining. That's the endomysium, lining around individual muscle cells. These connective tissues go beyond the muscles to form the tendon, right? So they extend down and form the tendon to attach us to bone. These are elastic, right? They can go back to their normal shape after stretching. And this is so that when our muscles contract, we have some sort of tension and we can pull on whatever body part, whatever bone is going to allow us to move. In terms of the blood and nerve supply, these are well innervated and have a significant blood supply. So capillaries are everywhere because we need a lot of gas exchange. Because these are metabolically, we'll say, expensive cells, because they're using so much ATP. That means they're using a lot of nutrients. It means they're producing a lot of waste. A lot of cellular respiration goes on there, so we need a lot of capillaries to service that. We have somatic motor neurons that are going to talk with these muscle cells so that we can cause contraction, right? And that each nerve, like in all of the skeletal muscle cells that it innervates, is called the motor unit. And the more fibers that are contacted, the larger the motor unit, the more force we can get. If we need fine detail, precise movements, then we have smaller motor units. So big movements that don't require a lot of finesse can have larger motor units. Smaller, more fine-tuned movements would use smaller motor units.